All right, welcome to part one of a series of very important lectures dedicated to skeletal muscle hypertrophy. We're going to go ahead and delve into the mechanisms of hypertrophy. In the next lecture, we'll sort of talk about the molecular basis of hypertrophy. So one of the things to know is that hypertrophy plays a role in nearly all sports. It's not just physique sports such as Arnold Schwarzenegger shown here to your left, or Aaron Stern to your right, but also almost any sport, including football, hockey, soccer, boxing. Skeletal muscle plays a major role. So one thing I want to talk to you about is that, of course, as you know, we have slow twitch muscle fibers, or ST, and fast twitch muscle fibers, or FT, in this graph. And Tesh did a really interesting study where if you look at powerlifters, which is what you see to the right, nearly 70% of their muscle fiber area was dictated by fast twitch muscle fiber hypertrophy. Now that indicates that their type of training preferentially hypertrophies the fast twitch, but not the slow twitch fibers. In contrast, bodybuilders actually had about 50% of their fiber size was dictated by the slow twitch fibers and 50% by the fast twitch. Now this indicates that bodybuilders non-preferentially hypertrophy both fibers. And it also shows that hypertrophy of both fibers becomes important for the physique athlete. Now Tesh followed this up with another study where he actually took the mean muscle fiber area, so individual fibers. He looked at a control non-trained group, bodybuilders, and powerlifters. What you notice is that powerlifters' individual muscle fibers are larger than the control group and bodybuilders. And in fact, bodybuilders were no different than the control. Now, this isn't necessarily run across the board for all studies, but this study in particular did find this. And what's important about that is this: the bodybuilder's overall muscle size was larger. Now, wait a second here. I'm going to say that again. The bodybuilder's overall muscle size was larger. But how can that be if their individual fibers were the same size? Well, the thought is that they were getting whole muscle hypertrophy via a process known as hyperplasia, which is the production of new muscle fibers. So it is possible that hardcore, brutal training can not only increase the size of the fiber, but it may also be increasing the number of the fibers. Remember, most of the studies we have that can actually count muscle fibers are going to be in an animal model, whereas in a human model, we're really projecting whether hyperplasia has occurred by counting small areas. So in nearly every animal study that we've done across the animal kingdom, we do see hyperplasia. And this is indicating that may happen in elite bodybuilders. Another way that we can hypertrophy is via sarcomerogenesis, or the addition of sarcomeres in series. So what you have here is when you see a lengthening contraction, you see that the 3 sarcomeres get lengthened. And we know we get a lot of damage from this. One of the adaptations from this is to add sarcomeres, which is in B. So a greater number of sarcomeres in series. So that's serial hypertrophy. So what is the stimulus for serial hypertrophy? It seems to be eccentric contractions. So in here, they basically had a decline running bout versus an incline running bout. The decline is going to cause much more eccentric overload or lengthening. This is the increase in sarcomeres that we see with the decline running, and that's the black group, as compared to the incline train group, which is the white group. Notice you have a higher amount of mean number of sarcomeres for the black group than the white group. And again, that's on the vertical axis. So when you're doing decline, more stretch, more sarcomeres. Why is that important? Well, we think it could maybe fill out the muscle near the insertion point. No, are you going to lengthen the total muscle? No. But if you add sarcomeres in series, it could make the muscle look fuller, such as when you do exercise like preacher curls or incline curls. Another method of hypertrophy is going to be an increase in cross-sectional area. That's when the muscle actually gets thicker. You're not just adding sarcomere serially. And there's two different types of hypertrophy that Zaszkiorski and Kramer propose. The first is sarcoplasmic hypertrophy, and that's hypertrophy of the sarcoplasm, an increase in glycogen, enzymes, water. And the next is myofibrillar, which would be hypertrophy via the addition of sarcomeres. So this is myofibrillar hypertrophy here. You can see that you have a myofiber beginning, and then you train, and you have radial myofiber hypertrophy. So meaning cross-sectional area is increasing by the addition of hypertrophy or hyperplasia of the myofibers themselves. This is a study that actually Schoenfeld was a part of in a lab out of Brazil, where you're looking at total body water after 16 weeks of hypertrophy training, and you see that it increases. Now, this may indicate that you're getting an increase in intracellular water, so which would be an indication of sort of the sarcoplasmic hypertrophy. Now, last thing I want to talk about in this lecture is the myonuclear theory of growth. So what's the myonuclear theory of growth? Well, if you look at the fiber under the microscope here, at the top left, you notice that it has numerous nuclei. And I circled one of these nuclei. Basically, we think that each nuclei controls a limited region of the cytoplasm, and that nuclei controls the protein synthesis of that muscle fiber. As you can see here, if we blow that up and we see the red fiber cross-sectional area with the yellow dot, that yellow dot represents the nucleus. When you lift weights, that increases protein synthesis, and that's going to expand the size of that what we call the myonuclear domain, which is the nucleus and the limited region of the cytoplasm it regulates. Now that can range from anywhere from 1,300 to 2,000 micrometers. But once you hit 2,000 micrometers, you've pretty much maxed out your potential for that nuclei. So you can't grow anymore unless you add more nuclei. And that's the myonuclear domain theory. So this is some research that I did back in my old lab. These are individual fibers. You see the arrows, what I'm pointing to, you see the fibers in cross-sectional area, and then what you see is basically nuclei on sort of cells which are dominated by the nuclei on the periphery of the cell. Those cells are called satellite cells because they're on the periphery of the cell. So basically we see this here. If you look at a muscle fiber, you have these satellite cells which are separate from the muscle but on the periphery. When you damage the fiber, those quiescent or resting satellite cells are then activated, okay? And so that's very important. You'll start to see that they expand and that they can actually end up binding to the cell and fusing to the cell. Here's an example of that. You have resting myofiber or a quiescent or resting satellite cell. There's myotrauma to the cell. The satellite cells then divide, which is, you see satellite cell activation and proliferation. They then migrate to the area that's injured, which is known as chemotaxis. They fuse to the damaged fiber, and that causes hypertrophy just by addition of their size, but also allows you for more hypertrophy because you have more nuclei. Also, those fibers can line up and form new myofibers, and we think that's a method of hyperplasia. So does this explain why some people grow more than others or hypertrophy more than others? Well, Marcus Baumann did a cluster analysis, and basically they did 16 weeks of knee extensor resistance training, and they found that out of the whole group of subjects, there were non-responders, which got 0% hypertrophy, moderate responders, which got 28% hypertrophy, and extreme responders, which got near 60% hypertrophy. Think about it. How many times have you seen the guy who goes in the gym and they just respond like crazy? That's actually true. And then you see the guy who goes to the gym, they don't make any gains. So, So that's very interesting. But why do you have these extreme responders? 60% increase in muscle fiber size. How the hell is that happening? Well, if we look here to the left, you see all the way to the left is the non-responders. In the middle of the graph is the moderate responders. On the right is the extreme responders. What you're seeing is mean fiber area. So you see the moderate responders hypertrophied a little, the extreme responders hypertrophied a lot. If you go to the middle graph, they're looking at myonuclear number, the amount of myonuclei, and what you see is that the non-responders didn't change, the moderate responders increased, and the extreme responders increased a lot more. And then if you look at myofiber area per myonucleus, you see the same trend all the way to the right. What explains the ability to increase nuclei per fiber? Well, you know, it's satellite cells, right? And again, this is pointing out here, non-responders, moderate responders, extreme responders, and again, myonuclear number, or myofiber area per myonucleus, the non-responders, moderate responders, extreme responders. If you look at satellite cells, we see the same thing. Clearly, the extreme responders get the greatest, most robust satellite cell activation, which explains the increase in myonuclei. So what's the mechanism of activation of satellite cells? Well, I mean, how many of you have heard of dystrophin, right? Most people. You've heard of muscular dystrophy. Why is it so important? Well, dystrophin is an extremely important complex. I'm pointing that to this here. Dystrophin is part of a complex known as the dystrophin glycoprotein complex. And essentially what this dystrophin complex and dystrophin in general, as you notice, there's a lot of different proteins in the dystrophin dystroglycan complex. But the dystrophin dystroglycan complex connects the contractile apparatus, which is the actin and myosin, or the filaments, essentially, and that connects that contractile apparatus to the cell membrane. So basically, it allows force transmitted from the myofibrils to be attached to the membrane. So the membrane's folding in along with the myofibrils. But also, what I want you to notice is this. There are a number of signaling molecules held within the dystrophin dystroglycan complex. One of them is right here. Point to that again. 1 second here. Neuronal nitric oxide synthase. So, or nNOS, so neuronal nitric oxide synthase. Like the name suggests, it synthesizes nitric oxide from arginine. So we see that here. Muscle contraction stimulates the conversion of arginine to nitric oxide by nNOS. So basically, that dystrophin dystroglycan complex senses that shear force. It stimulates nNOS that converts arginine to nitric oxide synthase. Nitric oxide stimulates the release of hepatogrowth factor, or HGF, and hepatogrowth factor, or HGF, activates satellite cells, which again are essential for muscle growth. But satellite cells can be activated by a number of growth factors. Testosterone can activate them. Nitric oxide can activate them. They can be activated by the nervous system, and numerous vascular growth factors can activate them as well. So this is just one possible method of activation of the satellite cells themselves. So in summary, hypertrophy can occur by several ways. It seems that bodybuilders hypertrophy both slow and fast. Powerlifters primarily fast. But it indicates that probably a wider repetition range probably will cause that, which we'll talk about later. We get serial hypertrophy. We can get cross-sectional hypertrophy. Cross-sectional can be sarcoplasmic or myofibrillar. And what allows us to have long-term hypertrophy is activation of satellite cells and ultimately more nuclei, which leads to the conclusion that expansion of the myonuclear domain increases via protein synthesis or breakdown, but we need new nuclei via satellite cell activation and differentiation. Thank you for your time, and I'll see you in the next lecture.